Hi, I'm Tricia Engalan, and for today's video, I am going to present and discuss the different major plant hormones. Plant hormone is a chemical produced in one tissue or organ and has its effect in another tissue or organ. Virtually, all life produces hormones to help coordinate and control growth, reproduction, and development. Therefore, these organic substances are highly valuable in a plant in order to thrive and survive. And there are 5 major hormones present in a plant, namely the auxin, gibberellin, cytokinins, ethylene, and abscisic acid. The first major plant hormone is the auxin. Auxin is considered as growth hormone discovered by Charles Darwin and his son in 1880. They found in shoot and root tips and promote cell division and stem and root growth. They can also drastically affect plant orientation by promoting cell division to one side of the plant in response to sunlight and gravity. The major functions of auxins is to promote phototrophism. A phototrophism is the ability of a plant to reorient the shoot growth towards the sunlight. Second, it promotes geotrophism. Third, it promotes apical dominance. Fourth, stimulate fruit development. And fifth, leaf abscission and fruit drop. And lastly, it promotes adventitious root development. So the picture shows how the process of shoot elongation is done. When sunlight is overhead, the IAA molecules or the auxin molecules produced by the apical meristem are distributed evenly in the shoot. Once the sunlight shines on the shoot at an angle, the IAA molecules move to the shade side and induce the elongation of cell on that side. And lastly, cell elongation results in the bending of the shoot towards the sunlight, and that is what we call the phototrophism. Therefore, auxins is a plant hormone that are responsible for shoot and root elongation. Second is the gibberellins. Gibberellins are also considered as growth hormones. It is produced mainly in developing leaves, shoot, root, and seeds. Also found seeds, young shoots, and roots. The major function of gibberellin is for stem elongations and to stimulate seed germination and flowering. And also, it helps for a fruit to have a growth. So, if a plant have less or no gibberellins, it will result dwarfism or it will not grow as expected. Therefore, gibberellins are important hormone of the plant in terms on its growing stage. Third is cytokinins. Cytokinins are plant-specific chemical messengers that plays a central role in the regulation of the plant cell cycle and numerous developmental processes. Cytokinins were discovered by F. Skoog and co-workers during the 1950s as factors that promote cell division. The word cyto means cell, while kinins means division. The cytokinins are responsible for creating new cell in plant or promote cell division. Cytokinins are also the cause that a plant creates a new branch. So without cytokinins, a branch growth will be impossible since a plant needs new cells. Also, cytokinins promote cell differentiations and delays senescence and aging. The fourth major plant hormone is the ethylene. Ethylene is also an important plant hormone and is used in agriculture to press ripe fruit. Ethylene hydrate is called ethanol. Basically, ethylene is a gas that causes ripen to a fruit. Like for example, you see a bunch of banana and one is already ripe. Then, that creates a lot of ethylene gas. And once that gas called ethylene is released, it accelerates the ripening of other fruit or banana, and they start releasing more ethylene gases, causing a chain reaction. More function of ethylene is it promotes seed germination, It involves in plant responses to pathogens and wounds. Lastly is the abscisic acid or the ABA. It is an important plant hormone involved in modulating responses to a number of abiotic stresses and also has a significant impact on plant defense against various pathogens. Functions of abscisic acid is to promote dormancy in woody stems, promote seed dormancy, and act as plant stress hormone. Basically, the abscisic acid is to prevent the growth of the plant, and the causes of wilting in a plant is due to high concentration of abscisic acid. Another function of this hormone is during drought, it ensures the stomata of the leaves is closed in order to prevent loss or evaporation of water.
Reproduction and germination. Germination, which is defined as the growth of embryonic tree contained within a seed. Seed germination depends on both internal and external conditions to support seedling growth. Inducing germination. Most important external factors to induce germination include temperature, water, oxygen, and sometimes light or darkness. Temperature affects cell metabolism and growth rates. Water is required for germination. Oxygen is required by the germinating seed for metabolism. Light or darkness can be an environmental trigger for germination and is a type of physiological dormancy.
Good day, I'm Tricia Aparente and I will discuss to you the seed germination, scarification, and breaking dormancy. Seed germination. All tree species can reproduce by seeds. Each species has a unique set of requirements for seed production and germination. The seed dispersal strategies vary widely from wind-driven seeds, such as on a maple tree, to seeds falling in the ground beneath the trees, such as a nut tree, or to seeds carried away by certain species of animals or birds, such as crabapple, being eaten by a bird and the seeds are dropped some distance from the tree. It determines continuous plant production necessary for human survival with regards to the supply of medicinal plants, feed for animal grazing, and food security. The propagation of some plants is only possible by seed, and if they are difficult to germinate, this threatens their continuous existence. Scarification. Some seeds have impermeable seed coats that prevent oxygen and water from entering the seed, causing a type of physical dormancy which is broken when the seed coat is worn away enough to allow gas exchange and water uptake the environment. Nicking, breaking, softening, or other ways weakening of the seed coating meant to speed up germination. It is important to reduce the hard seed content and improve germination for successful stand establishment of these crops. Breaking dormancy. The seeds of many species do not germinate immediately after exposure to conditions generally favorable for plant growth, but require a breaking of dormancy, which may be associated with change in the seed coats or with the state of embryo itself. Often involves changes in membranes initiated by certain hormones, notably abscisic acid, which inhibits germination, and gibberellin, which ends seed dormancy. Hi everyone, my name is Cedro. Today I'm going to report the roots. Roots. First to emerge from the seed coat is the primary root or radicle as it is called in the embryo. This radicle is composed of one major root that is thicker as its base and tapers towards the growing tip. Early root growth is one of the functions of the apical meristem located near the tip of the root. The meristem cells more or less continuously divide, producing more meristem root cap cells. These are sacrificed to protect the meristem and undifferentiated root cells. After the embryonic seedlings emerges from the seed coat and starts growing roots and leaves, the embryonic seedlings' food reserves are quickly exhausted. Most tree species grow a taproot from the seeds until moisture or an obstacle is reached, and the seedlings can then focus on spreading structural roots, absorbing water and nutrients, and letting the leaves manufacture food. So, tree root generally grow in any direction where are the correct environment of air, mineral, nutrients, and water exist to meet the tree's needs. Roots will stop growing towards on dry or poor. Let's proceed to the vegetative reproduction. Besides seeds, trees can be propagated with the assistance of mankind. Cultivars. Cultivars are propagated by grafting a bud or stem of the parent tree onto a seedling's root stock or the species. So cultivars is an organism, and especially one of the agricultural or horticultural variety of strain. Clones are propagated by encouraging roots to develop on cuttings of the parent tree. So clones or cloning is a technique to scientists use to make exact genetic copies of living things. Genes, cells, tissues, and even whole animals can all be cloned, and some clones already exist in nature. Sprouts and suckers. Start with dormant buds that come alive to form new shoots of the parent trees. Sprouts and shoots from the trunks of a tree. Suckers are shoots that originate from bands on the root systems. Both sprouts and suckers are the potential to grow into new, full zytrees genetically identical to the parent. Oftentimes, sprouts and suckers will not grow until the parent tree dies or becomes very sick or suffers from a severe injury. The buds are held in dormancy by hormones produced in the leaves. When these hormone levels drops below a certain point because of increased light level, as the old tree thins its branches and leaves, the dormant buds begin to grow. Sprouts and suckers are the shoots that develop on the trunk and branches of trees, while suckers are shoots that grow from roots or the base of a tree. Vegetative layering. It is a common form of vegetative propagation when branches or stems come in contact with the soil. The cambium tissue sometimes will form roots. In this way, former branches might become trunks or of several new trees genetically identical to the parent. It is also a technique of plant propagation where the new plant remains at least partially attached to the mother plant while forming new roots. Tissue culture. The last one is tissue culture. It is an invention of modern science to exact cells of desirable trees and growth these cells into trees within laboratory conditions. Trees from this process are genetically identical to the plant. It is also the growth of tissues or cells in an artificial medium separate from the parent organisms. This technique is also called as micropropagation. And that would be all. Thank you.
Tree root physiology. Root physiology is ability of the roots to sense the environment and adjust accordingly. Roots develop entirely rather than from buds that occur on stems and the parts of the tree lob above the ground. Root parts. Root cap, this part of the root is of its very tip. The cap protects the tip of the root as it pours through the soil by the elongating root tissue behind it. Apical meristem. This part of the root provides the cell from the root. Cap in front and for the region of elongation behind this area. The sensor will cause the apical meristem to change direction of growth to avoid an obstacle and its path. Region of elongation. Cells in their fixed positions elongate to reach mature dimensions in the region of elongation. It was water pressure to push again the walls of cells to stretch the cellulose fibers in the new cells to make them elongate and push the cap through the soil. Region of root hairs. It is in this area where cells develop a more mature from a change into the epidermis and the cortex. Rhizosphere. The thin layer of the soil that sticks to the root is called the rhizosphere. It is the zone about 1 millimeter in width surrounding epidermis of the living root hairs that contains million more soil microorganisms than anywhere else in the soil. Mycorrhizae. Mycorrhizae is beneficial fungi tree. Mycorrhizal fungi are naturally occurring fungi that prepare the soil in the forest. Mycorrhizae are not exactly a root and they are not exactly the fungus. They fall halfway between. They are important because they form symbiotic relationship with the tree's root and act as extension of the roots.
Tree trunk physiology. The trunk supports the crown and holds the leaves up to the light to allow photosynthesis to occur. The trunk is a highly ordered arrangement of living, dying, and dead cells. The roots are bathed in a nutrient-rich liquid which transports this nutrient plus moisture to the top of the tree where it's all consumed during photosynthesis. The tree trunk has to expand in diameter as the tree grows in its search for moisture and sunlight. A tree, as it says, supports an ever-flowing root system that must be maintained at all times. If the process fails to provide water at any point, the tree will eventually die due to the failure of both water and food requirements that are necessary for life. Outer bark. The outer bark is a layer of dead cells which protects and covers the layer of living cells of the cambium. Bark is a tree's natural armor and protects it from external threats. Bark also has several physical functions. One is disposing the tree of waste by absorbing them and locking them into its dead cells and resins in the bark. The bark also influences light passage. It regulates gas exchange. It recognizes and defends against pathogens. It controls tree temperature and many other things that contribute to the survival of the tree. The outer bark is the tree's protection from the outside world. Continually renewed from within, it helps keep out moisture and the rain and prevents the trees from losing moisture when the rain is dry. It insulates against cold and heat and wards off insect enemies. Inner bark or core cambium. The tissue occupies the space between the outer bark and the phloem tissues and is called the core cambium layer. This is the layer that generates new bark. Inner bark or core cambium. This is the layer that generates new bark. In roots, the cambium tissue may store sugars or starches to fuel the spring sap flow. In leaves, the cambium tissue is the mesopolysis doing the photosynthesis. The inner bark is a pipeline through which food is passed to the rest of the tree. It lives for only a short time, then dies and turns to cork to become part of the protective outer bark. It consists of secondary phloem tissue whose innermost layer conveys food from the leaves to the rest of the plant. Phloem. Phloem next to the inner bark develops from the outside layer of the cambium and is the food tract to the roots. Phloem carries large quantities of manufactured food or sugar that are transported from leaves toward roots. Phloem carries large quantities of manufactured food or sugars that are transported from leaves toward roots. When the tree is healthy and growing and sugars are abundant, the extra sugars are stored in the form of starch, which can be converted back into sugars and moved by the phloem to where it is needed in the tree. Cambium. The cambium and its zone is a cell generator, reproductive tissue called growth meristem that produces both the inner bark cells of the phloem and new living wood cells in the xylem. The cambium is a watery layer, only a few cells thick. The cambium is the growing part of the trunk. It annually produces new bark and new wood in response to hormones that pass down through the phloem with food from the leaves. These hormones, called auxins, stimulate growth in cells. Auxins are produced by leaf buds at the ends of branches as soon as they started growing in spring.
Apical dominance. Apical dominance is the phenomenon where the main central stem of the tree is dominant over side branches. The apical bud subjugates all other side branches by producing growth hormones that reach and affect all parts of tree. Those hormones regulate side branches just grow ever outward, and as a result they typically have a wide angles of attachment to the stem. Because of this, the apical bud of the is the only one grows consistently upward or towards the light, while the remaining branches are cast in a supportive role by not being allowed to compete for upward growth.
Xylem is living softwood and is located inside the cambial zone and next to the heartwood. Xylem carries water substances dissolved in water and mineral from the roots to the leaves. The outer position of xylem is conducting and storing starch in the same class. Plus, it conducts water and substances dissolved in water to the leaves. The major structure for the water transport into xylem are called vessels. In angiosperm, heartwood, woods, and trachids in the gymnosperm conifers. Heartwood, the inner portion of the xylem, is non-conducting wood that stores starch and is sometimes called the heartwood. Most of the heartwood did woody tissue and serves as repository for many waste products of the produced in the tree's living tissue. It also functions to support the weight of the crown. The cells produced in the spring wood when the tree's rapid growth. Later in the season, the cells produced are smaller, thicker walled, and slower growing quickly are wide to allow them to carry the large amounts of the water needed for the tree's rapid growth. Later in the season, the cells produced are smaller, thicker walled, and slower growing and called summer wood. Spring wood is the light in color and fast-growing wood. This difference in the cells seen in a cross-section of the trunk as oryx are typically in the temperature forest trees, and tropical humid rainforest trees grow continually and do not have rings. The oldest portion of the tree is the bottom and the innermost rings on the trunk. Symplast is the network of living cells, and the connection between living cell starch is stored in the symplast, axial parenchyma, ray parenchyma, sleeve tubes, companion cells, cambium, and plasmodesmata all make up the symplast. Vessels and tracheids. Vessels in hardwoods and tracheids in conifers also conduct water and substances dissolved in the water. Vessels are vertically aligned tubes made up of the cells that transport liquid. Vessels are found only in angiosperm. Tracheids are dead single-celled pipes that act much like vessel but are only found in gymnosperm.